Good day everyone, I'm Maria Bonita Makinay Busayong, a licensed professional teacher and a MAED student major in science education at Basilan State College. One of our requirements in SciEd 204 is a video presentation discussing some tools used in genetic engineering. But before that, let us first define what is genetic engineering. Genetic engineering is the process of using recombinant DNA technology to alter the genetic makeup of an organism. Traditionally, humans have manipulated genomes indirectly by controlling, breeding, and selecting offspring with desired traits. But how genetic engineering stands, let us explore the tools used in genetic engineering. Tools used in genetic engineering is the polymerase change reaction or the PCR. This technique is used in molecular biology to amplify a single copy or a few copies of a segment of DNA across several orders of magnitude, generating thousands to millions of copies of a particular DNA sequence. Second tools used in genetic engineering is the restriction enzymes or the molecular scissors. These are enzymes that create one incision on each of the two strands of DNA at specific locations based on the nucleotide sequence. So they are also called as DNA cutting enzymes because their task is to cut DNA into strands. Tools used in genetic engineering is the gel electrophoresis. This is used for various purposes, from viewing cut DNA to detecting DNA inserts and knockouts. It is also used to estimate the molecular weight of a protein and nucleic acids, purifications of isolated proteins, monitoring changes of protein content in body fluids, and blotting applications, and many more. So it appears to be as a monitor when doing the genetic engineering. The fourth tools used in genetic engineering is the DNA ligase. These are enzymes that can create covalent bonds between nucleotide chains. So DNA ligase are also called as the glue enzymes because they are making connection. The fifth tools used in genetic engineering is the polymerases. These are collectively referred as the groups of enzymes that catalyze nucleic acid molecule synthesis. So when we say catalyze, it is either speed up the reaction or slow down the reaction. Six tools used in genetic engineering is the prokaryotic host. These are able to multiply their plasmids along with foreign DNA, also multiply to produce millions of copies, referred to as a colony or in a short clone. The seven tools used in genetic engineering is the eukaryotic host. These are prepared to produce human proteins since these hosts with complex structures with distinct organelles are more suitable to synthesize complex proteins. 8 tools used in genetic engineering are the selection of small self-replicating DNA. Small circular pieces of DNA that are not part of a bacterial genome but are capable of self-replication are known as plasmids. Plasmids are often used as vectors to transport genes between microorganisms, so they serve as the transport materials. The last tools used in genetic engineering is the methods to select transgenic organisms. So the main tools here are the transgenic organisms to be used. We have to choose what type of transgenic organisms to be used depending on the procedure that we will be doing. Let us recall them again. So the first tools is polymerase change reaction or the PCR. Second is the restriction enzymes or the molecular scissors. Third, the gel electrophoresis. Fourth, is the DNA ligase. The 5th is the polymerases. 6 is the prokaryotic host. 7, the eukaryotic host. 8 are the selection of small self-replicating DNA. And the last is the methods to select transgenic organisms. Okay, so that is our topic for today. I hope you are going to join me again. So once again, I'm Maria Bonita Makinay Busayong. Thank you and God bless.